The popular story of Barbara McClintock is a comfortable, familiar myth. We are told she was an obscure underdog, a lone genius whose discovery of jumping genes was ignored purely because of her gender. But this narrative hides a much more unsettling historical truth. When McClintock presented her defining evidence in 1951, she was not a marginalized outsider. She was one of the most elite, heavily credentialed geneticists alive. In 1944, she was elected to the National Academy of Sciences. The very next year, she became the president of the Genetic Society of America. Her peers rejected her data not because she was unknown, but because it broke the fundamental laws of biology. Her theory that genes could move was considered impossible. Yet today, in early 2026, researchers have proven her exact mechanism physically reconfigures the 3D architecture of human DNA, acting as the primary engine driving cancer. To understand why the scientific establishment refused to accept empirical, visual proof from a researcher they already revered, we have to look at the rigid rules of the mid-20th century genome. In the 1940s, genetics operated under a strict structural dogma. Genes were universally understood to be static, immovable entities. They were described in textbooks as beads on a string, permanently locked into linear sequences along the chromosome. Mutations were viewed exclusively as rare, permanent chemical accidents triggered by external forces like radiation or toxins. The idea that a genome could contain natural, internal regulatory movements was completely absent from the scientific consensus. McClintock shattered that assumption. By tracking the modeled color patterns of corn kernels and applying specialized chemical stains, she found a way to physically map the structure of chromosomes through a light microscope. While systemic sexism absolutely restricted her early career stability, forcing her to rely on rolling research grants rather than securing a comfortable, tenured university position, her intellectual authority among geneticists who understood her methods was virtually unmatched. It was her rigorous mathematical crosses and cytological slides that provided the definitive proof. She isolated a controlling element she called dissociation, noting regular, visible breakage points on Maize chromosome 9. But dissociation didn't just break the chromosome. McClintock observed that it physically transposed. It jumped to entirely new locations, shutting down pigment genes wherever it landed and turning them back on when it moved away. Decades before DNA sequencing even existed, McClintock empirically proved that the genome was not a passive string of beads. She showed it was a dynamic, highly sensitive organ that actively rearranged itself in response to environmental stress. The problem was the gap in technology. Eminent scientists simply could not follow her highly idiosyncratic mathematical logic. Without a known molecular mechanism to explain how DNA could literally cut and paste itself, her purely visual proofs seemed completely biologically impossible. The establishment largely dismissed her jumping genes as a bizarre anomaly unique to corn. She was met with stunned, impenetrable silence at her 1951 Cold Spring Harbor presentation. But her discovery wasn't a quirk of a single plant. It was the foundational mechanism of genomic regulation. To see exactly how she measured the impossible, we have to go back to her Cold Spring Harbor laboratory and look directly at the physical evidence left in her earliest Maize slides. In the mid-1940s, Barbara McClintock worked in near isolation at her Cold Spring Harbor laboratory, surrounded by thousands of harvested ears of Zea maize. Committing her career to corn was a heavy professional trade-off when most ambitious geneticists favored fast-breeding organisms like fruit flies. Maize was an agricultural crop with a long breeding cycle and immense genetic complexity. Many considered it too unwieldy for pure genetics. Yet, it offered one distinct advantage. Its macroscopic traits, like kernel pigment, provided a highly visible generational record of microscopic genetic events. To understand the magnitude of what she found in those pigment records, we first have to understand the rigid, unbreakable rule of mid-century biology she was about to shatter. It was universally known as the beads on a string model. Throughout the 1930s and 1940s, long before the discovery of the DNA double helix, the scientific consensus held that chromosomes were static, linear structures. Genes were viewed as fixed, immovable entities permanently anchored to specific physical coordinates. Under this central dogma, a gene could not naturally change its location. Mutations were understood strictly as permanent chemical mistakes, usually triggered by external forces like radiation or toxins, which damaged the gene precisely where it sat. McClintock understood the mechanics of this static model better than almost anyone alive. In 1931, she and Harriet Creighton published a landmark paper in the Proceedings of the National Academy of Sciences that revolutionized cytogenetics. Titled A Correlation of Cytological and Genetical Crossing Over in Zea Maize, their empirical evidence proved that chromosomes physically exchange material during cellular reproduction. This 1931 publication alone secured her legacy as a master of physical chromosomal mapping. Because she had literally mapped the architectural rules for how chromosomes behave, she recognized immediately when those rules began to break down. Between 1944 and 1948, a bizarre physical signal emerged in her carefully controlled breeding lines. The anomaly manifested as erratic, unpredictable patches of color on the maize kernels. A common historical misconception is that she was simply observing pretty modeled corn. In reality, she was tracking highly precise mathematical disruptions in inheritance. She documented thousands of kernels under a microscope, logging the exact ratios of purple, brown, and colorless sectors. The pigment genes were inexplicably turning on and off repeatedly during the cellular development of a single kernel. To her contemporaries, this heavy variegation was nothing more than an unstable mutation rate. The establishment assumed the local pigment genes were simply experiencing rapid, localized chemical damage, creating a chaotic phenotype that wasn't worth deeper investigation. But McClintock's solitary, exhaustive concentration over thousands of individual specimens revealed a hidden regulatory logic beneath the chaos. The timing and frequency of the color changes were far too deliberate to be random chemical errors. Her calculated ratios indicated that the pigment genes themselves were perfectly intact, but their expression was being systematically interrupted. Something inside the maize genome was physically moving from one location to another to shut the pigment off. She knew the corn was fundamentally breaking the accepted laws of genetics. But to convince an establishment that believed genes were locked tightly in place, she couldn't just calculate the mathematical anomaly. She had to build a way to actually see it. In the late 1940s, seeing a specific genetic mechanism in action was considered a physical impossibility. The structure of the DNA double helix was entirely unknown, and chromosomes were largely viewed as amorphous, microscopic blurs that only condensed during cell division. To map the origin of the modeled patterns she observed in her maize crops, McClintock could not rely on the high-throughput sequencing we use today. She had to locate specific structural landmarks on maize chromosome 9 using only chemical stains and a standard light microscope. This approach demanded an extreme, almost punishing level of cytological discipline. She refined custom carmine staining techniques, applying specific chemical washes to make the faint chromosomal structures visibly distinct during the Pachytene stage of meiosis, when chromosomes pair up. It was a brutal, physically exhausting process. She prepared thousands of fragile glass slides by hand, subjecting herself to severe eye strain and grueling hours alone at the bench.
She was relying entirely on her own highly trained visual acuity to separate true biological architecture from random optical noise. Her surviving laboratory notebooks, now preserved as essential historical artifacts, reveal the exact physical records of this labor. They are filled with precise, hand-drawn sketches mapping out microscopic features, identifying specific chromosome knobs, centromeres, and tails with absolute geometric consistency. Through this relentless, tedious repetition, she documented a recurring, highly specific structural anomaly. At a precise genetic locus on chromosome 9, the physical body of the chromosome was quite literally breaking in half during cell division. At the time, the prevailing biological dogma dictated that genes were rigidly locked in stable, linear sequences. If a chromosome broke, most geneticists of the era viewed it as random cellular damage, perhaps a fatal mutation triggered by natural radiation or environmental decay. This created a sharp epistemic boundary between her observations and what the scientific establishment was prepared to accept. Skeptics could easily argue that these microscopic fractures were merely chemical artifacts, unnatural distortions caused by her own harsh carmine scanning process rather than native genomic events. But McClintock's statistical breeding data cross-referenced flawlessly with her visual maps. When a mature kernel exhibited a specific sudden loss of pigment, the stained cells from that exact genetic lineage consistently displayed the precise structural break on chromosome 9. She named this specific breakage point dissociation, or DS. Her meticulous correlations proved that this was not a random tear in the genetic fabric. It was a reliable, targeted structural failure that directly controlled the expression of the kernel's color. Yet, this is exactly where her methodology hit the absolute limits of mid-century technology. Her visual evidence was fundamentally restricted by the resolving power of 1940s optics. She could clearly see the catastrophic breakage, but she could not observe the underlying molecular cut-and-paste action causing it. Without a biochemical framework to explain how genetic material could actively slice itself apart, her purely cytological proofs seemed mechanically impossible to her peers. She was documenting a profound biological truth, but she lacked the molecular vocabulary that would only arrive decades later. Despite this missing mechanical layer, her hand-drawn maps of the Maize genome remain some of the most accurate ever produced using light microscopy. She had successfully isolated the exact physical location of the genetic fracture, proving that chromosomes were not immune to structural alteration. She had found the reliable location of the break, but as she tracked the D's locus across successive, carefully bred generations of maize, she watched the impossible happen. The break itself moved. The microscopic break didn't just vanish, it relocated. By 1948, McClintock was tracking a specific, fragile spot on Maize chromosome 9. She named this precise location dissociation, or DS. The prevailing dogma insisted that chromosomes were rigid structures and mutations were permanent chemical accidents. But under her microscope, McClintock saw that DS was physically cutting itself out of the chromosome and inserting itself somewhere else. She crossbred thousands of plants to track where it went. When the DS element jumped into the middle of a gene responsible for the kernel's dark purple pigment, it acted exactly like a blown fuse. It shut the pigment gene off completely. As the corn kernel grew, that specific lineage of cells remained permanently pale, creating a distinct colorless sector on the otherwise purple surface. But the most shocking part was the reversal. If DS jumped back out of the pigment gene later in the kernel's cellular development, the genetic circuit reconnected. The pigment gene immediately turned back on, creating a sudden, tiny burst of color. The modeling she had been studying wasn't a random chemical breakdown. It was a physical, visual record of a gene moving in real time. Yet McClintock's data revealed something even more radical. The D's element couldn't move on its own. It remained entirely frozen, locked in place, unless a second, distinct element was also present in the genome. She named this second element activator, or AC. The activator functioned as an autonomous master switch. It could dictate exactly when the D's element would jump and how frequently it would break the chromosome. This was a staggering conceptual leap. McClintock hadn't just discovered that genetic material could physically travel. She had discovered that the genome was not a passive library, but a dynamic, self-regulating machine. It requires immense intellectual courage to compile data that violates every established rule of your discipline and still choose to trust the evidence over the consensus. In 1950, she formalized her proof in the Proceedings of the National Academy of Sciences. The paper, titled The Origin and Behavior of Mutable Loci in Maize, laid out the ACD's system using exhaustive, mathematically precise breeding ratios. Her cytological mapping empirically proved the genetic movement, but here lay the profound vulnerability of her work. She had absolutely no way to explain the biochemical mechanism behind it. In 1950, the double helix structure of DNA hadn't even been published. Without a molecular framework to explain how a solid chromosome could chemically splice itself open and stitch itself back together, her purely mathematical proofs seemed biologically impossible. To the wider scientific establishment, her exhaustive datasets looked like an elaborate illusion. Most peers assumed she was misinterpreting standard recombination events or looking at a bizarre anomaly that existed only in corn. But McClintock knew what she was looking at. She had the physical specimens, the stained microscope slides, and the predictive models to back every single claim. Armed with her data, she prepared to present her findings at the elite Cold Spring Harbor Symposium in the summer of 1951. But when she laid out her meticulous cytogenetic evidence, the response from the world's top biologists was devastating. In the summer of 1951, McClintock stepped up to the podium at the Cold Spring Harbor Symposium on Quantitative Biology. She was there to present the ACDS control system to the most elite minds in genetics. The official 1951 symposium program lists her presentation simply as chromosome organization and genic expression. She walked the audience through years of meticulous cytological data, correlating visible chromosome breakages with complex Mendelian breeding ratios to prove these elements were physically jumping. She expected rigorous, high-level debate from her peers. Instead, she was met with a profound, stunned silence. Historical testimonies from the audience that day don't describe mockery or open hostility. They described total, overwhelming bewilderment. The scientific establishment didn't dismiss her data because she was an obscure outsider. They dismissed it because they literally lacked the conceptual vocabulary to process what she was showing them. The gap between her classical methods and their molecular assumptions was too vast. Remember, in 1951, the double helix structure of DNA had not yet been published. The absolute, unquestioned law of early 20th century biology was that chromosomes were rigid, stable architectures. Genes were thought to be permanent beads locked onto a string. For McClintock's transposons to be real, the genome had to be capable of cutting and pasting its own code. At the time, mutations were strictly understood as permanent chemical damage from radiation. Without a biochemical mechanism to explain how DNA could move, her theory seemed impossible. Even the few scientists who could follow her incredibly dense statistical crosses found a way to rationalize the data away. They categorized her findings as the Maize anomaly, a bizarre, hyper-localized quirk unique to corn and entirely irrelevant to human biology. The psychological weight of this moment is rarely captured in the textbooks. It is the profound isolation of holding airtight, empirical proof that you are right, while realizing that your entire scientific generation is conceptually blind to it. She was speaking a language no one else knew. Faced with a wall of incomprehension, McClintock made a calculated decision. Archival publication records show that by 1953, she deliberately stopped publishing detailed accounts of her transposon research in major journals. She chose to self-censor. This wasn't a surrender, but a strategy for professional survival. By halting her public push for the jumping gene theory, she avoided further alienation. She quietly protected her funding, her fields, and her position at the Cold Spring Harbor Laboratory. She continued her breeding experiments in the background, filling notebook after notebook with undeniable patterns of inheritance. She knew her statistical evidence was structurally sound, and she recognized that molecular biologists would eventually have to discover the mechanism for themselves. She essentially placed her greatest discovery in a time capsule. It would take nearly 20 years and the maturation of molecular biology for researchers to finally find the exact biochemical mechanisms she had predicted using only corn kernels in a microscope. But when that molecular proof finally arrived, McClintock's Maize anomaly wouldn't just be vindicated as a quirk of botany. It was about to become the master key to understanding human evolution, genetic disease, and the hidden engine of the genome itself. The silence of 1951 didn't last forever, but the scale of the truth was bigger than anyone dared to imagine. For decades, the extra DNA McClintock tracked was dismissed as junk, a biological byproduct of evolution. But as our sequencing tools reached single-molecule precision, we realized those jumping genes weren't a quirk. In the human genome, transposable elements make up roughly 50% of our code. In her beloved Maize, that figure climbs to nearly 90%. We aren't a static blueprint. We are a shifting mosaic of elements that have the power to build, regulate, and occasionally destroy. This brings us to the present. In January 2026, at the Plant and Animal Genome Conference, researchers presented the first comprehensive fiber-seq maps of the Maize genome. Using this long-read technology, they visualized chromatin accessibility at a resolution McClintock could only dream of with her light microscope. The data was a direct vindication of her 1940s hypothesis. The study identified over 100,000 accessible regulatory regions, doubling the known regulatory landscape. Crucially, nearly half of these master switches are located directly within transposons, proving they are the primary controllers of gene expression. But the impact of McClintock's controlling elements extends far beyond the cornfield and into the frontier of human medicine. In March 2026, clinical data using the TE single analytical framework began mapping how these jumping genes influence the progression of Parkinson's disease. We now see that in the dopaminergic neurons of the brain, specific families of transposons like lines and endogenous retroviruses are not silent. When the cell's epigenetic brakes fail, these elements awaken, triggering a viral-like immune response that drives the neurodegeneration defining the disease. We see a similar pattern in cancer dedifferentiation. As aggressive tumors lose their specialized identity, they do so by hijacking the same mobile machinery McClintock first observed in a New York cornfield. The movement of DNA is the very mechanism that allows a cell to reinvent itself. Barbara McClintock called them controlling elements because she saw the genome as a sensitive organ of the cell. She believed it could rearrange its own architecture in response to environmental stress. Today, we call this field epigenetics, and it is the foundation of modern biology. In 1983, 32 years after her peers met her data with silence, the Nobel Committee finally caught up. Barbara McClintock became the first woman to receive an unshared Nobel Prize in physiology or medicine. It was the ultimate acknowledgement that her vision had outpaced her era. She didn't seek the spotlight. She simply kept looking through the lens until she saw the truth. Her data wasn't an anomaly in corn, it was a preview of the dynamic logic that builds every living thing on Earth. She proved that the code is not a fixed script. Today, we no longer view our DNA as a rigid string of beads. We see it as a fluctuating, living engine that uses its own history to innovate and adapt. McClintock showed us that the most important parts of the code aren't the ones that stay still. The next time you look at the vibrant colors of an Indian corn kernel or read about a breakthrough in gene therapy, remember the woman who saw the impossible. The genome is not a finished book. It is a story that is still being written, one jump at a time. Her legacy is a reminder that, in science, the most dismissed anomalies are often the keys to the next revolution. If the data shows you something the rules say is impossible, it might just mean the rules are wrong. Trust the evidence over the dogma. Barbara McClintock didn't just find jumping genes, she found the pulse of life itself. And in 2026, we are finally learning how to read the message she decoded 80 years ago. Stay curious, and we'll see you under the microscope next time.